Happy Monday. Monday. I have a song I'm writing. It's called Hyundai, Hyundai. You can probably figure out the tune it goes to. Hope everybody had a good weekend. Anybody remember it? All right, enough silliness. We spent some time last time talking about techniques, and today I'm going to spend some additional time talking about some new techniques, some of which I think you're going to find very medically relevant. So hopefully you'll find this interesting, what I have to say. So last time as I was talking, I finished up discussing polyacrylamide gel electrophoresis. And more specifically, I talked about SDS page. And SDS page is what we use to separate proteins by electrophoresis. And those proteins we have to manipulate so that they behave like DNA molecules. So we manipulate them so they behave like DNA molecules. And the manipulation that we did was that we treated them with SDS. The SDS coated those proteins. It made them lay out flat like rods. And that coating gave them a very negative charge. And the longer that rod was, the more negative charge it would have. A common question I get, and I got it at the end of the lecture last time, was, well, does that negative charge kind of negate any other charge that's in that protein? And the answer is yes, it does, because there's a lot more of those SDSs on there than there are charged residues within that protein. So that effectively makes that a polyanionic substance, meaning multiple negative charges. And it's not a perfect technique. Okay? Some proteins behave a little anomalously, but the vast majority of proteins behave very well in SDS. And we can see their molecular sizes. So the next figure I want to show you is an example of a protein gel where there are, on both sides, this is what's called a ladder. And a ladder simply is a set of proteins of known size that are there. And then we can see a protein being purified, various steps of the purification process, various quantities of that protein being present, and we can see an estimated size from that. As noted last time, this technique really isn't useful for isolating a protein that we want to use later. It's more of an analytical technique that tells us how much protein do I have here, what's its size, and how pure is it compared to the things I'm trying to purify it away from. So that's what SDS page is very, very useful for. SDS page, as we shall see, is very useful when combined with another technique that I'm just getting ready to talk to you about. And so I'll do that in just a moment. Before I do that, I'll remind you of the structure of SDS, and no, you don't have to draw SDS, but this is what it looks like. It's a long-chain fatty acid. These are all carbons and hydrogens down here. And at the end, we've got a sulfate. That sulfate is what gives it the negative charge that is necessary for the electrophoresis. Okay. Well, as I said, if we combine SDS with, SDS page with another technique, we get a really, really powerful technique. And it's a powerful technique for studying things like cancer, for studying how drugs work, and so forth. And so I want to spend a few minutes talking about that, because I think it's a pretty cool and fascinating technique. Okay. So the technique that I want to discuss first is called isoelectric focusing. Isoelectric focusing. All right? Now, this technique uses some substances that are interesting in their structure. We don't have to really talk too much about them. But suffice it to say that it relies on a mixture of compounds that have varying amounts of charge. Some of them are very, very, very negative, and some of them are very, very, very positive, and many of them are in between. So we can imagine, for example, that we might have a compound that might have 40 negative charges, another compound in our mixture that might have 39, 38, 37, 36, and then on the other side we might have something that has 40 positive charges and 39, 38, 37. And so we've got a complete spectrum of charges that are in this mixture that I have. Okay? Well, if I take this mixture and I treat it very carefully, and I put it into a tube, kind of like the columns that we've been talking about, and apply an electrical field to that tube, what will happen is that the things that are the most negatively charged will move towards the positive electrode, and the things that are the most positively charged will move towards the negative electrode. And if I look at that tube when I have finished doing this electrical treatment of the tube, what I will see is that the things that had 40 minus will be the closest to the end, followed by the 39 minus, the 38 minus, the 37 minus, et cetera, et cetera. And in the very middle, I'll see that's where things had a zero charge. Make sense? Okay. So this tube turns out to be really nice because if I take this tube and instead of just putting these compounds in there, what if I put my mixture of proteins in there as well? Will they separate according to those charges that are in there? And the answer is yes, they will. So the proteins that are the very most negatively charged will be the closest to the positive electrode, and those that are the most positively charged will be closest to the negative electrode. And I'll see a spectrum of proteins across that range. Make sense? Now these tubes have a sort of a gelatinous-like material in them. All right? So what I've done is I've separated these guys on the basis of charge. And in essence, what I've done is I've separated them on the basis of their pI. We don't need to worry too much about that at the moment, but that's basically what we've done. Okay? Those that have the lowest pIs on one side, those that have the highest pIs on the other side. All right. Now, why do I tell you that? I tell you that because, first of all, that technique is a way of separating things by their pIs. But more importantly, when I combine that technique with SDS Page, I get something really, really cool. All right? So let's imagine we do what I just said. We take that tube, we take our proteins, we mix them, we run that electrical current, and so we separate guys on the basis of their pI. The most negative on one side, the most positive on the other side, and the others a mixture in between, right? Let's say I very carefully take that tube and I open the tube up and I take that gelatinous material out and I lay it on top of a gel where I'm going to do SDS Page. Okay? So I add SDS to this material that's in here, so I coat it all with negative charges. It's not starting out with negative charges because I haven't put anything on it to start with. I just have the proteins by themselves. I add the SDS to it, and then I run the material on that tube downwards and separate on the basis of size. What I've just done is something we describe as two-dimensional gel electrophoresis. 2 dimensions. The first dimension separated on the basis of size. On the basis of pI. The second dimension separated on the basis of size. Now this figure schematically shows you what I just described. Here are some proteins that have been separated on the basis of their pI on the top, and then we've applied them, we've added SDS, and we've applied them to an SDS Page gel, and separated them on the basis of, first on the basis of pI, second on the basis of their size. All right, so what do we see? When we see this gel, if we were to look in the upper left, we would see the proteins up in here. We would see the proteins that have the most negative charge and are the largest. Down here, we would see the ones that have the most negative charge and are the smallest. Down here, the most neutral charge and the smallest, etc. You get the idea as we go across this two-dimensional gel. Okay? Well, this turns out to be an extraordinarily powerful analytical tool as well. Okay? Let me show you what an actual 2D gel looks like. Okay? An actual 2D gel looks something like this. And you say, wow, what a mess, right? What a mess this thing is. Well, it turns out this mess is pretty darn cool. Because this mess, in this case, was taken from all of the proteins of a given cell. I can separate all the proteins, and I can decide, using a variety of techniques, which protein corresponds to which spot. Every protein will have its own unique spot. All right? That's one thing. Second, this type of analysis, if you're very, very careful, is very reproducible. It's very reproducible. So what does this mean? Well, let's imagine I want to study a certain cancer, and I want to understand something about that cancer. I might take, let's say I've got liver cancer. I might take some cells, some DNA that's from my liver that are not cancerous, and I might separate those proteins on a 2D gel. And then I might take that liver tumor that I have, and I might take those proteins and apply them to a different 2D gel. And then I would compare the 2D gels and say, which proteins are different? Which proteins are being made in the cancer but not being made in the regular cell? Which proteins are being made in the regular cell that are not being made in the cancer cell? Which proteins are being made more in one than the other, or less than one in the other? All kinds of things I can ask. But now, as a result of this analysis, I can understand the complete picture of every protein in a normal cell versus a cancer cell. I'm not restricted to cancer. I could say, well, what happens? I've got this new drug that I want to treat people with, and I want to understand what effect this drug has on our cells. I take one batch of cells over here, don't treat it with the drug. Another batch of cells over here, treat it with the drug, do 2D gel electrophoresis, and then ask the question, which proteins are different? That's really powerful, extraordinarily powerful tool. Okay? This type of analysis tells us very quickly how we're affecting the expression of proteins inside of cells. And since proteins are the workhorses of cells, we know very, very quickly something about the cancer, something about the treatment, something about your cells versus my cells. Maybe I'm making more of one protein than your cells are. Neener, right? Okay. If that's a bad protein, then you're going to go neener, right? So, all right. But you get the idea. So 2D gel electrophoresis is a very, very powerful tool. Questions on that? Yes, up top. Very, very good question. His question is, if I take a protein that has quaternary structure and I treat it with SDS, will the subunits come apart? We'll take a vote. How many people think they will come apart? How many people think they won't come apart? Okay? Majority rules. Majority's right. The subunits will come apart. Okay? And there's two reasons. One is that hydrophobic interactions can, in some cases, help stabilize quaternary structure. But even if the quaternary structure is not being stabilized by those interactions, when the protein, the individual subunits themselves unfold, it disrupts completely the interactions between the subunits. So quaternary structure is completely disrupted by SDS treatment. Yes? Okay. Okay. No, actually, that's not true. So what he said was that if it denatures, then the individual strands are going to run faster than the entire protein. But remember, the same happens in any kind of denaturation. So when I know I've got a multi-subunit protein, I know what the individual subunits are. I do not know what that overall structure is. Now, your question is very good, because if we want to understand the quaternary structure, we have to use other kinds of analytical techniques to understand that. Sometimes we can be working with a protein and have no idea it's a dimer, for example. Okay? So if we just simply do SDS page, we won't know that we've got a dimer of two identical subunits. All right? We have to use other analytical techniques to do that. But you're exactly right. Okay? Good questions. Yeah? Yeah. Are you going to have to tell me which spot is which? Is that what you're saying? Well, I'm not going to say which protein is that spot, if that's the question. All right? But I might point to something on here and say, where would you find, for example, the largest, most positive proteins? Right? That would be fair. Yeah. Yeah. Okay. Says here, vertical, does gravity have an effect? The answer is no, it does not. And the reason it does not is because the force, the electromotive force that's driving through there is far greater than any gravitational effect that it would have. Yeah. Okay? Good questions. Okay. So that's 2D gel electrophoresis. Kind of cool stuff there. Here's a comparison of a normal colon cells versus a tumor. And you can see here's a protein that's made in fairly small amounts in a normal colon, and in the tumor it's made in very abundant amounts. You can also see there's probably a difference right here and here as well. So there's some differences that we see between them. And there are some very powerful computer tools that will do that analysis and say, how much darker is this spot than that spot? So we can get some pretty good ideas for that. Okay. Well, hopefully what I've told you last time is that, and hopefully you remember, is that we have to do many steps in purification to get a protein that's ultimately very, very pure. And you might wonder, well, why do we care so much about getting it very, very pure? If it's just fairly pure, is that okay? For some analyses, fairly pure is okay. For many analyses, it's not. Okay? So one of the analyses that we commonly want to do when we purify a protein is we want to determine its structure. Remember, we can't predict it from the computer. So we actually have to determine it. And to determine it, we have to have absolutely pure protein. Techniques that allow us to do that, I'll briefly mention in a little bit, include nuclear magnetic resonance and X-ray crystallography. And I'll say something about those in just a little bit. Okay? So those two techniques really, really require us to have things that are very, very pure. And we can see on this gel this analytical use of SDS page to help us sort of follow what's happening during the purification process. Here's, the material we got out of the cell. Here's after we use salt fractionation. That's something I haven't talked about here. You're not responsible for, but it's another purification step. Here's some ion exchange chromatography. Here's what happened after we did gel. And then finally, after we did affinity, we got something that looked like this. So we can see our protein started out over here, and we finally got it to a place where it was fairly pure. One of the things that we need to do during that purification process is to follow it. We have to follow it. We have to test at each step, where's my protein at? How much of it do I have? Okay? Because if I don't do that, I really won't have any idea of how much material I'm working with. Further, I ultimately might like to go back into my cells and determine, well, how much material is in a given cell? How many copies of that protein are found in a given cell? So doing the kinds of analyses that we see here in Table 3.1 are very commonly done by researchers. And they're done partly also so they don't throw away the baby with the bathwater. Remember? We're doing a step, let's say centrifugation, where we have a precipitate and we have the supernatant. If we think that the material we want is in the pellet and all we keep is the pellet and we throw away the supernatant, that water might have had the baby in it, right? So we don't want to do that. We've got to check everything and make sure it's there. Well, this scheme shows a purification going through the steps that you saw on the last figure. And this step in the purification follows the amount of protein we've got. So we started out with a protein where we were able to determine we had 15,000 milligrams. That's 15 grams of protein. That's a lot of protein. And after we fractionated it by salt, we ended up with 4,600. Okay? Well, you might say, well, why do we lose protein? Well, we lose protein because we're throwing away things we don't want and hopefully keeping things that we do want. That's what we saw in the last gel. In fact, we saw some of those disappearing. After ion exchange chromatography, we had 1,278 milligrams. After gel filtration, we had 68.8. And by the very end, we had 1.75. But you recall from that gel, about the only thing that was on that gel was, in fact, our protein. So out of that whole mixture, I got 1.75 milligrams of protein. Okay? Now, I have to follow the protein. I have to measure its activity. All right? So that protein most commonly might be an enzyme. How much product does it produce per time? That would be a measure of its activity. And so I can measure its activity as in some unit that I decide on what that is. This 15,000 milligrams of protein had 150,000 units of activity to start with. Okay? This protein, or this mixture that I had to start with, had a specific activity of 10 units per milligram. Simply the number of units divided by the number of milligrams. Okay? The yield I had was 100% because I haven't done anything yet. I haven't thrown anything away. And my purification level was 1 because, again, I haven't done anything to it. By the second step, what I see is that I've reduced the total protein by a considerable amount. I've reduced the protein by more than a factor of a third. By more than a factor, well, it's less than a third of what I started with. But I still have a pretty good amount of the units. Now you'll say, well, how come I don't keep all the units? Well, no purification technique allows you to keep everything. You're going to lose some as you're going along. So this is pretty good retention. We can see, in fact, well, how much retention was it? Well, it was 92%. It was equal to the number of units I had here divided by the number of units I started with. That's 92%. We can see that the result of my purification helped to increase the specific activity because the specific activity went from 10 units per milligram up here to 30 units per milligram up here. Remember, this is the units divided by the total milligrams. So I've increased the specific activity by a factor of 3, and my purification, therefore, has increased by a factor of 3. If I go all the way down, I see I get less and less material, and I still lose material. By the very end, I've got 1/3 of my total activity, but look how much junk I've gotten rid of. An awful lot of stuff I don't want. So I've actually done pretty well right here. My retention, my yield was 35%, which was this number, 52,500, divided by the 150,000, of course, times 100 to get percent. And I got 35% yield. So that was pretty good. My specific activity went through the roof. It went from 10 here up to 30,000. 30,000 was units divided by total protein again. So my purification level went up 3,000. I purified this guy 3,000-fold. Okay? This is a pretty cool purification that I've done. And each step along the way gave me purer and purer protein until I finally got down here to the very end. Okay, questions about that? Yes, Omar. I'm sorry? How do you quantify purification level? Purification level is equal to the purification that you have at any given step divided by what it was when you started. Okay? So it's this 30,000 divided by 10, which is what gives me my 3,000. Okay? Yes? So would that be a pretty typical yield? Is that a pretty typical yield? It's going to vary a lot from protein to protein. I'd say this is a pretty good yield, yeah. Other questions? Okay, good. All right. We're going to move from purification to some characterization. I'm going to skip over a few things in characterization. There are some techniques that your book goes through that are really very old and nobody does anymore. So I don't think there's a reason we should mess with them. All right. One of the things I do want to talk about are what are called polypeptide cleavage agents. Okay. Polypeptide cleavage agents are substances, whether they're chemical or enzymes, that can break proteins into pieces. It can break proteins into pieces. You might wonder why you want to do that, and the answer to that question is because some of the analytical techniques don't work very well on great big proteins, but they work nicely on smaller pieces. We'll talk about one in just a little bit. Okay? So being able to break proteins into specific smaller pieces is really helpful. And as I said, there are some of these compounds that work chemically, some that work enzymatically. I will tell you about both, some in both categories. One of the more common ones that's used for chemical cleavage is a compound called cyanogen bromide. And what cyanogen bromide will do is it will break a protein on the carboxyl side of methionine residues. So we find a methionine in a protein, and we look on the carboxyl side of it. That's where that cyanogen bromide is going to break it. Okay? I'm not going to discuss the other chemical agents, which means, again, I'm not going to hold you responsible for those. But I will talk about some of the enzyme ones, because some of the enzyme ones we'll talk about later in the class. Trypsin is one of those. Trypsin is one of the very easy ones to remember. It works on the carboxyl side of lysine and arginine residues. So it's an enzyme. It's actually an enzyme from our digestive system. And that enzyme in our digestive system breaks proteins in the carboxyl and lysine residues. Okay? Another one we'll talk about later in the term is thrombin. Thrombin is involved in the blood clotting process. And thrombin cuts on the carboxyl side of arginine. Now, if I look it up here, you say, well, the carboxyl side of lysine and arginine residues, what do those two have in common? Anybody remember? What's that? They have an NH3 plus on their side chain. Okay? And it turns out that is, in fact, what they're looking at as they are doing their cleavage. Okay? All these enzymes are examining the R group side chain of the amino acids and then cutting appropriately. All right? Chymotrypsin is one we'll talk about. I'm not going to hold you responsible for the specific amino acids, but I will note that these tend to be fairly nonpolar, and in this case, they tend to be fairly large side chains. And we'll talk a little bit more about that later. The last one I'll mention is carboxypeptidase A, and it's a very easy one to remember. It's the only one that cuts on the amino side, and it acts like a Pac-Man. You guys are too young to remember what Pac-Man was like, but Pac-Man, of course, started at one end, and he just started chewing in. Well, that's what this guy does. It starts at the carboxyl end, and it chews in one amino acid at a time. Okay? Now, it says it doesn't work on arginine, lysine, and proline. It actually doesn't work very well on those, but for our purposes, we'll say it works on all of them, just to make it easy for you. So it starts at the carboxy end, and it just starts chewing. If I leave the carboxypeptidase A in the presence of a protein, it will completely eat the protein down to amino acids. And you're probably thinking, will it eat itself? Will it eat other carboxypeptidase A's? And the answer is yes, it will. So proteases all have that property. They don't have any distinction between is this a protease or not a protease and act on it. They will eat up each other as well. Yes? Do these proteins become charged after these reactions? Their charges will not change from what they were, but we can imagine, let's say we had a protein that had a charge of +7. I might cut it into a protein that has a +4 and a +3, for example, depending upon where I cut it. So depending upon where I cut it, I'll make subunits that, smaller pieces that might have individual charges, but that total charge is going to be the same. Okay? Okay. Let's see. We talked about this guy before. I didn't show you the reaction for it, but I talked about DTT lecture before last. And DTT, I will remind you, is a reagent that will reduce disulfide bonds to sulfhydryls. It works like mercaptoethanol does. In fact, it works identical to the way mercaptoethanol does. It just starts out with a different structure. But the end result is the same. We get sulfhydryls where we started with disulfides. And we see that the dithiothiotol starts as disulfides and ends, it starts out as sulfhydryls and ends up as disulfides. So DTT works like mercaptoethanol does. And now I want to spend a little time before I do that, I'll talk about, obviously you guys know the genetic code is the information inside of cells that tells the cell what sequence of amino acids to put together to make a protein. And that information is encoded ultimately in the DNA. The DNA that's converted into messenger RNA. We'll talk about that next term. But suffice it to say that it is actually way easier to determine the sequence of a protein by sequencing the DNA that codes for it than it is to try to find the actual amino acid sequence by breaking down a protein and then analyzing each one of those amino acids. All right? That was the way they used to determine the sequence of a protein. And that's some of those older techniques I was talking about that we're not going to discuss. Okay? They're very tedious. They're very time-consuming. To give you an idea, to determine the amino acid sequence by analyzing a protein, amino acid by amino acid, might take a month for one protein. Okay? The way our DNA sequencing technology is today, we can actually determine the entire genome of an organism, if it's a bacterium, in a day. That would include several thousand proteins in one day. So DNA sequencing really has taken over our analysis of proteins. Okay. So what I want to turn our attention to at the moment is a set of immunological techniques that we use to analyze proteins. And they're used in combination with SDS-PAGE. Okay? So what I'm going to describe is something called Western blotting. I'm going to come to that in a second and show you. But before I talk about Western blotting, I need to say a little bit about the tool we use to perform the analysis. All right? The tools that we use to perform the analysis in Western blotting are antibodies. And antibodies are proteins of the immune system. They have a very useful property in that they're designed to bind to things. They're designed to bind to structures. And more specifically, they're designed to bind to specific structures. So let's imagine for a moment I am studying the protein insulin. Okay? Insulin is a hormone that's found in our body. And I want to analyze this protein by Western blotting, which is what we're getting ready to do. In order to do that, I would have to have an antibody that recognized and bound to insulin. I'd have to have an antibody that recognized and bound to insulin. Turns out I can make one of those. Or more specifically, an organism can make one of those for me. How does that occur? Well, let's say I have a bunch of, let's say, human insulin that I want to study. I take that human insulin and I inject it into a bunny rabbit. Our immune system is set up so that it recognizes invaders. And when it recognizes invaders, it synthesizes antibodies that bind to those invaders. Since bunny rabbits don't have human insulin, they have bunny insulin. They see the human insulin as an invader, and they start making gobs of antibodies. I got a flu shot last week. Somebody shot into my arm a whole bunch of proteins from a flu virus. And my immune system is right now recognizing those and making antibodies against those, so when a real flu virus comes along, it's going to bind to it and keep me from getting infected. Well, this bunny rabbit that's making my insulin does this for a while, and then I say, okay, well, I could really use those antibodies. It turns out to be very easy. I take a little bit of the bunny rabbit's blood, and then in that blood contains these antibodies that bind to insulin. I can purify those antibodies, and now I've got a very powerful tool. These antibodies will bind to insulin, and usually only to insulin, specifically human insulin. Okay? That's step one. Right? Now, we don't have to worry about the various nomenclatures, antigens, and blah, blah, blah. Antigen is simply what an antibody binds to. All right? And this is what the structure schematically of an antibody looks like. We don't even need to worry about that too much. All right? That's really what it looks like more realistically instead of schematically. Okay? Here is an antigen or antibody binding to an antigen. All right? So here's that protein that the antibody recognizes, and you can see it binding to it here. Okay? Well, I tell you that because this is essential for something we call a Western blot. Why do I want to do a Western blot? Well, a Western blot allows me to not only separate proteins, but also identify in that separation which protein is the one I'm interested in studying. Okay? Let's go through the steps here. So I got a mixture of proteins. I bust open some cells. I say, okay, I'm interested in knowing how much human insulin is in these cells. Or is there human insulin in these cells? Okay? So I take these proteins. I don't purify them. I just separate them on an SDS page system. All right? I then take that gel that I have just done, and I lay it on top of a membrane. That membrane is specially designed to bind to those proteins that are in that gel. So what I can do is use an electric field and transfer them out of the gel and onto the membrane. So now they become stuck to the membrane, and that membrane has all the proteins lined up just like they were in the original gel. Everybody with me? I can take that membrane then and put it into a little Ziploc bag, and yes, we do use Ziploc bags to do these sorts of things. Treat it with some buffer and with an antibody that's been made against that insulin. Okay? Well, you can imagine what's going to happen. The place where insulin is present in this mixture is where the antibody is going to bind, and the other places which don't have insulin it's not going to bind to. I take out that membrane. I wash it off to get rid of the loose stuff that isn't bound to anything. And then I treat it with something that tells me where are there antibodies. This can be a color reagent. This can be a light reagent. Sometimes we make things flash. It doesn't really matter what the reagent is. But we're simply asking the question, where do I find antibodies? I get a color, I get a spot, I get whatever, but now I can see where in that original mixture my protein was. Excuse me. I get an indication of how much I might have. And consequently, I've learned something about insulin in those cells that I was studying. This is true. I can do this for essentially any protein that I want, so long as I have an antibody that I make against it. Yes, question? Boy, that's a very good question. How does that antibody still recognize that protein if I've treated it with SDS? Okay? First of all, it doesn't always. There are some times the treatment of SDS will disrupt that and not let that occur. But the treatments that I'm doing here will, A, partly get rid of the SDS, and second, the antibodies will oftentimes recognize primary sequence, not tertiary structure. So if it recognizes short segments of sequence, those are always present in the protein. And as long as I can expose them and let the antibody get at them, it will recognize them. It's not perfect. It won't always do that. Sometimes you'll do it and it just won't show up. But most of the time, in fact, it will. Good question. Other questions? Clear as mud? Anybody awake? All right. Pop quiz then, right? No. Okay. All right. The next thing I want to talk about, these are all cool techniques. And in fact, as I'm going along, we're seeing these techniques are getting more and more sophisticated. And they're actually getting, in many cases, newer as well. All right? What I want to talk about that has really revolutionized our ability to analyze proteins in recent years is a technique called MALDI-TOF mass spectrometry. There's a mouthful of a name. Okay? MALDI-TOF has a longer name than I'm going to go through here. All right? But it's a technique that involves proteins within an evacuated chamber that are accelerated by an electrical field. Wow, there's a mind-boggling kind of thing for you to think about. What does it look like? What do we do? All right? That's not what it looks like. That's what the result looks like. Okay? And that's what the blah, blah. Oh, I'm missing my link. Oh, blast it. What did I do? All right, so I'm going to have to describe it to you. All right. You guys get to visualize, right? So visual learners like to visualize. So you guys are going to be visual learners. You're going to visualize this. Let's imagine I have a protein that I want to determine the molecular weight of. Okay? Protein's a pretty big thing, right? Protein might have a molecular weight of 200,000. And I told you some techniques don't work real well on large proteins, so I got to break them into pieces. So I might take a piece of a protein and analyze the molecular weight of that piece. Maybe it's got a weight of 8,000, much easier for me to handle. How would I determine what its mass is? Well, MALDI-TOF allows me to do that surprisingly precisely. Okay? Surprisingly precisely. How do I do it? Well, I take my protein and I put it into a little crystal. And I put it like on the head of a pin. All right? You with me so far? I got my protein, it's in a crystal, it's in the head of a pin. This crystal I can volatilize very easily with a laser. Meaning I shoot it with a laser, it turns into vapor very quickly. Okay? Well, if the crystal turns into vapor, you can imagine what happens to the protein. It's no longer bound by the crystal. Right? Okay. So I've got this crystal, I've got this protein, it's on the head of a pin, and now I take this head of a pin and I stick it into an evacuated chamber, meaning it's a vacuum. Everybody envision that? So I got this long tube, I've put my stuff in at one end. Here in there is the head of a pin, it's got this, this crystal and it's got my protein in there. All right? Then, this tube is set up. A, it has no air in it, so it's completely evacuated. B, at the other end, there's an electrical plate. meaning that I can generate an electric field in here. Okay? 3, it's got a laser in there. So here's what I do. I shoot the pinhead with the laser. The material volatilizes, and that's time 0. When the laser hits the pin, I've got time 0. I turn on the electrical field. What happens when the laser hits the material? The protein volatilizes. It's in the chamber now, floating. Well, the protein is going to be charged. It turns out that ionizing a protein, when I shoot the laser at it, it causes the protein to gain a charge. It gets a charge. It starts out with 0 charge. It's gaining a charge. All right? My electrical field, now I've got a negative over here. I've got a positive over here. Guess what's going to happen? It's going to be pulled to the negative by the positive-negative interaction. It turns out that the time it takes to move from where it starts to hitting the plate is related to its molecular weight. The MALDI-TOF, when I talk about MALDI-TOF, the TOF stands for time of flight. How long does it take to go from here over to here? Okay? Each time it has a charge of +1. Let's say I have something that has a molecular weight of 8,000 and it has a charge of +1. It's going to move at a certain rate, right? Let's say I compare that to something that has a weight of 4,000 and a charge of +1. Which one's going to move faster? The smaller one. The weight to mass ratio is smaller, so therefore the smaller one will move twice as fast. If it had 16,000, it would move half as fast. Yes? We can set it up so it has a positive or a negative, so we don't really even need to worry about that. But for our purposes, we'll say it has a positive. Okay? All right. So now what's happened is by measuring the time it takes to do this, I can very precisely measure the molecular weight. And I can even do more really cool things with that because it turns out that when I hit it with the laser, several things happen to it. One of the things that happens to it is that big 8,000 molecular weight piece doesn't stay 8,000 molecular weight. It frequently breaks into smaller and smaller and smaller pieces. If I measure the mass of each one of those pieces, I can actually determine the sequence of the protein. How so? Well, here's a protein that has a sequence of glutamic acid, glycine, methionine, arginine. If I measure the whole thing, it's got a mass of 621. If I lose one amino acid from this end, I lose a glutamic acid. I know the weight of the glutamic acid that's lost. The difference between this and this is glutamic acid, and only glutamic acid will have that mass. I know it had a glutamic acid. Next, it loses another one. Next, it loses a glycine. Next, it loses a methionine. You get the idea. I can look at the weight of all those fragments that are in there and say, oh, here's the sequence of my protein. It's a really cool way and a really fast way to determine the sequence of a protein if that's what I need to do. Now, you might think, well, that's kind of tedious. It is very tedious. It's computer intensive, very computer intensive. However, if it's set up properly, I can determine the sequence of 4,000 proteins a day using this technique. That's why I'm not telling you about the old stuff. 4,000 proteins a day using this technique because the computer just does it automatically. There's a little robot that'll punch. I'll take a 2D gel, and I'll take one of those spots, and a little robot will take this spot and put it in the mass spec, and then this spot and put it in the mass spec, and then this spot and put it in the mass spec, each time determining the sequence of each one as it goes along. Really cool technique. So MALDI-TOF allows us to do some phenomenal analysis of molecular weight. Questions about that? Everybody's kind of blown away by that. You guys look tired today. Should we do a song and then finish early? I just had a feeling. I don't know. I just had this feeling about that. Okay? Okay. This is a song. I see people leaving. That's interesting because people who are leaving probably don't want to have extra credit questions, I'm guessing. Oh. Okay. There goes your voice. You're losing your voice. So, this is a song I wrote for BB350, but you can substitute 450 and it'll still work, okay? It's called This Song's for BB450. It's 1 o'clock and Ahern's talking, Henderson and Hasselbach and PKAs and buffers I should know. This song's for BB450. I hope that maybe he'll think the way we wrote our answers wasn't crazy. I really need the partial credit, so this song's for BB450. It's really groovy that it improves me watching lectures and QuickTime movies. I really need to go and download those podcasts for BB450. I'm feeling manic, I'm in a panic. I'd better study my old organic. It has reactions that I need to know. This song's for BB450. I know he said it, that's why I dread it, 'cause I skip Fridays, extra credit. 'Twill probably haunt me, that lowly 0 grade in BB450. It could be steric or esoteric, that carbons get so anomeric. I'm too hysteric, better let it go. This song's for BB450. See you guys on Wednesday.